And then we'll finish up with this part of the course by talking about a little bit of molecular biology, and then we will finish up with the last slide, which is covers just about everything we've talked about so far. It's going to talk about cells, proteins, how genes are regulated, and I'll use as that topic how bourbon is made. So you'll get a little of that. Now, proteins are composed of amino acids, which you probably know, and proteins do a lot of different things. If you think back to the cell membrane, we had all these different kind of transporters, and these transporters are all proteins. You know, we have carrier proteins. There are structural proteins. I will be sending you out a link to an article to read about extensin, which is a protein that's found in the cell wall. And there are proteins that help stabilize the membranes. There are proteins that help stabilize the cell wall. There are a lot of proteins. You guys know that the ones that we're going to spend most of our time on, which are enzymes, which actually carry out reactions. They do something. They take a substrate and they rip it apart into two different things, or they take two different things and stick them together. They do a variety of things. Every reaction is catalyzed by an enzyme. You've all heard of receptor proteins, and plants have a lot of receptor proteins. We have a lot of receptor proteins. And you've heard about, you know, when you hear about receptor proteins, what do you think about first? In humans, what do you think about? Where do we talk about receptor proteins? Taste. Nervous system, brain. You know, that what we find more and more is that, like, you know, you've heard of like there are these receptors for drugs. And what we find really is that most of these compounds don't actually get into the cell. They bind to an integral membrane protein and cause something to be released inside the cell. Okay? There are proteins that regulate gene expression. There are hundreds of proteins that are needed for a piece of RNA to be made or for DNA to be replicated. Now, usually in number 6, the terminology, usually when we had the term AS or the extension ASC, so protease breaks down protein. What does invertase break down? Sugar, sucrose. Why is it called invertase? Because it was invert before we knew it was sucrose, so it's invertase. It takes a sucrose molecule and it breaks it down to glucose and fructose. Okay? So you take a disaccharide, which is a glucose and fructose molecule with a covalent bond, and the invertase breaks that bond. And just as an aside, that's how you know, you get these chocolate-covered cherries that have all that liquid inside. That liquid actually starts out as sucrose. And when they make those things, they put a little invertase in there, and the invertase breaks that sucrose down. It becomes a lot thicker and fills it up with that you know, that sort of that what are you going to call it, the syrup that's inside. Because it's easier, cheaper to do it than it is to put that syrup inside them. Now, let's see if I can actually get this to work. Okay.
They change it every.
Now, let me see if I can get one. Yeah, it changes. I mean, it's the citric acid cycle. It's been a couple of Okay, what does that mean? Well, metabolic pathways. Am I going to be able to show this to you or not? Ah, good. Here we go. Okay, here we go. This is a little bit of every dot you see here. Come on. Okay. I won't touch it. Every dot you see here is an enzyme. And this is a small part of the pathway. You just mentioned the TCA cycle or the Krebs cycle. This is the Krebs cycle right here. Okay? So my point is that every single thing that's made in the cell, every single compound that has a phosphate added to it or has something added to it is catalyzed by an enzyme. So there are literally just thousands and thousands of enzymes. This small part of the pathway, the green, I think, is what you find in plants. This is what you find in anything, bacteria, animals, or plants. And you've probably gone, if you know, like when I was in grad school, I had this big metabolic pathway chart on the wall in my office. Okay. Now we can just go to it online. And it's pretty neat because you can find something, you can say, well, I found this metabolite in the cell. How can it get there? So you can punch in the name of that metabolite, and then you can find all the possible reactions that can produce it. And then you can start building hypothetical pathways. So if you find something that's in the cell, you can now much easier sort of narrow in on what it might do. Did anybody see 60 Minutes last night? There was a kid, 15-year-old, that came up with a biomarker for pancreatic cancer. And that's basically, he knew this compound was there, and so he was able to figure out what he could react it with to make it do something that could be measured. 15 years old. Pretty good. So the point here is that each of those little dots is a very specific enzyme. Protein. Enzyme in this case, actually. Okay. Let me close it, hopefully. Close all tabs. Wow, it worked. Okay. This is what the basic building block looks like. This is an amino acid. Now, this is where if you have to brush up on your organic chemistry, there's some links on one of the pages. You need to understand what these structures are. And what we have is a carboxylic acid on one side and an amine on the other side. And so this is the carboxylic acid part, this is the amine, and then this is the alpha carbon. So what we're going to see is that this is the building block. So I would take this second amino acid and I would put it over here. And then very importantly is we have this R, and an R can be anything from a proton to a methyl group to a very, very complex number of carbons. Okay? You can have a phenol ring hooked on as an R group. Then here's just a series of them. Glycine is the simplest amino acid. There are some proteins that are nonpolar. There are proteins that are aromatic, and you should be able to recognize these. Don't worry about knowing the names of the proteins, but obviously from organic you should be able to understand what the characteristics of these individual proteins are going to, these amino acids are going to be. And let's see if I get lucky enough to open up two. Wow, how about that? Here's their arranged in terms of their functional groups. The R, remember, is the functional group. So we have some basic and we have some acidic amino acids. And then we have some, these are the most common amino acids that we find. There are many more than this, but these are what you're going to run into in most living organisms. Now, what kind of reaction is this? Y'all should know this. You've all had organic chemistry. What? Dehydration reaction. What's a dehydration reaction? You look at the little colors there. What's happening? You're taking water out. You're taking a CO, a carboxyl group from one side and a proton from the other side, and you're sticking them together, okay? And I didn't bring my sheets of paper down. I'll do that Wednesday. What you have there then is you lose water, and now you have what that is called a peptide bond. Now we have taken two amino acids and stuck them together. Now we have what's called a peptide. If you have two of them together, it's a peptide, and you can have 1,000 of them together, and it's a peptide. It's a polypeptide, okay? Now, the bottom there, we're seeing just the linear arrangement of the amino acids, and let me go to the next one, which is a sort of, what we're trying to show here in 2 dimensions is the three-dimensionality of the molecule. One thing that you should get out of this course that you usually don't have before you come in is the fact that things are three-dimensional. And what's really cool about this is that this peptide bond is fairly straight. Okay? It's like a sheet of paper, and next Wednesday I'll bring some paper down and show you. And then you have these strings of amino acids strung together. Okay? And the bright green is the R groups. And the R groups are going to determine the characteristics, the solubility characteristics of that protein, right? So if all those R groups are nonpolar, what happens when you put the protein in water? Is water polar or not? Come on, folks. What's going to happen if you put a nonpolar something in water? What's going to happen? It's going to separate out, okay? Now, in reality, most proteins are polar and nonpolar. They have domains, and we'll talk about that later. So they have portions where you have a lot of polar groups that are together. Then you have portions where they're nonpolar groups together. Okay? And we'll talk about this again. I'll pull up a picture of a membrane again. So this repeating unit does not change. CCNCC does not change. It is the R groups that change and provide the characteristics of what this chain's going to be. Okay? A really good representation of a lot of proteins is a telephone cord. And and we're going to talk about the levels of structure, but a telephone cord we don't have many of those anymore. It's fairly characteristic of, if you took a telephone cord and wadded it up in a very specific way, then and you could see it. That's what a lot of proteins look like. Okay, and of course if you stretched it all out, would be the individual amino acids strung together. Questions? So we have all these amino acids. We get about 20, a few more. You know, we make most of them, but there's some amino acids that we have to consume. That's why it's hard to be a good vegetarian, because it's hard to get all the amino acids that you need and all the vitamins you need. You have to be careful, you know. There's really not many things that actually have all the amino acids in them. You know, that's why we have to eat a variety of things. Now, let's go back to the telephone cord, and let's talk about the 4 levels of structure of proteins. And let's take that telephone cord and let's, take a magic marker and put dots, pull the cord out till it's completely straight, and paint a lot of dots on the cord. Each of those dots would be an amino acid. And what the primary structure is, it's determined by the order of the amino acids. So if we, and this will be a little more apparent, we're going to touch on protein synthesis Wednesday or Friday. This was amino acid 1, this is amino acid 2, this is amino acid 3, this is amino acid 4, this is amino acid 5. I'm not going to go on. You get my meaning, that the primary structure is the order of the amino acids. Glycine, methionine, whatever, cysteine. It's how they're put in. Now, if you think about that, and most proteins are hundreds of amino acids long. You could see, excuse me. You could see then, and I'm getting ahead of myself, but let me go ahead and talk about this. Let's say these are all polar, and these are all polar. Then if we take this, these polar and polars are going to tend to try to get close to each other. Okay? Teleologically, the polar will want to be with the water, and the nonpolar will want to be away from the water. So you can start seeing this thing, take that phone cord, and now let it go so that you have a spiral. And we're going to talk about the next order. Okay? Questions? Secondary. Here's the phone cord, right here. And this is called a helix, an alpha helix. A phone cord is a helix. And the other formation that it can take is basically a sheet, a flat, more or less, protein. Now, what causes this? What causes this to fold up this way? Right, mostly the R groups and the R groups that are close to each other. Okay? So let's say I have a protein that goes across the bottom of the room. The secondary structure would be maybe 2 feet from the side because of the R groups in those first amino acids interacting with each other. And so remember, the R groups are going to cause that. They're not going to break the bond, but they're going to cause it to swing around a little bit, right? So you might have 20 amino acids out from the wall. Let's say that's 2 feet, and that might form a spiral, an alpha helix. You could go over in the center of the room, and because of the interactions between the amino acids that are in the center of the room, they might interact to form a sheet. And it's very common for a single protein to have all these structures in the same protein. Okay? So usually what I like to think of as the secondary level of protein, it's due to the interactions with closely located adjacent amino acids within a small space. And a lot of times we call that space a domain, D-O-M-A-I-N. Now here in the corner, you see a protein, you see this cartoon that has the sheets, and also this is supposed to be a helix, okay? So here's a single protein that has all these characteristics. Now, let's go up to the next level, and this is the interaction between domains. Okay? So what does that mean? That means if I go to the right side of this room and I come out 2 feet, I say this is nonpolar. Okay? Then I go to the opposite side of the room and come out 2 feet and say this is not polar. This is nonpolar. And then I go to the center and say the center 2 feet is nonpolar. That's 3 different domains. And if I let that protein go in water, those 3 nonpolar regions are going to try to get together. Does that make sense? And then that produces the phone cord when you've gone from the spiral to the wadded up. So the tertiary structure is this interaction between the separate domains. And that gives you then the final three-dimensional structure of a single peptide. So if you take that phone cord and you fold it up the exact same way every time, that unique 3-dimension would be an enzyme or a protein. And then I take the phone cord and I fold it up in an entirely different way. That's a different protein. It's going to be dependent upon the R groups, and the R groups are going to determine the secondary structure, and then which will be a domain, and then the tertiary structure is going to be those domains, interactions between those domains. Okay? Questions? Yes, ma'am. R groups can be polar or non-polar. Okay. An R group can be anything from a from a hydrogen to a carbon to several carbons to a ring. So the R groups are going to make the protein basic, acidic, neutral, polar, non-polar. Okay. Don't worry, you don't need to look at a protein and say, "Oh, that's basic." You should understand what these different groups are, though, right? Okay. Yes, ma'am. I'm sorry? The domains are based on the polarity of adjacent, in the same neighborhood, of the amino acids in the same neighborhood, okay? The tertiary is between distant neighborhoods. So we've taken the phone cord and now we've folded that phone cord up into, wadded it up into, but we don't wad it up. It folds the same way every time. Every single protein is unique. Different amino acids, different 3-dimensional shapes. And that's 3-dimensionality which determines what it does, okay? Now, so let me go to this one because I'll show you. One thing you notice here is a chain, and I will try it. I've not had much luck with actually showing these in real time. But you see this is a chain, and this is a chain. So now we can go to quaternary, which is the exact same graphic, and that's between the interactions between these 2 polypeptides. So if I go back to this one, see it doesn't change. Here is one peptide, and here is another peptide. Okay? So quaternary, and I make this point, is when you have it's the interaction between 2 different polypeptide chains, between 2 proteins. And guess what? Most enzymes have at least 2 subunits. Sometimes they're identical, sometimes they're not. Think back to, we talked about the rosette. that rosette of proteins where cellulose is synthesized, remember? Each of those rosettes is a protein. Each component of that is a protein, okay? There are proteins that are composed of 20 different polypeptides. Okay? So the quaternary is interaction between at least two polypeptide chains. So in the tertiary, we're just talking about one of these chains. And in this case, this chain and the interactions between the same polypeptide, as opposed here, now we're looking at these two different polypeptides. Okay? Just to finish up with this cartoon, the top is the primary structure, and this is from your book. The A1 then is just showing you the primary structure, the repeating backbone, and then the next one down is showing you the spiral from the effects of the closely linked amino acids. And then C, you're seeing this thing fold up into a three-dimensional shape. And then in D, you're seeing two different polypeptides that are coming together to form, in this case, what would be an active enzyme. Okay? Are the two chains from the same protein? They can be, and they can be different ones. So if you think about what has happened during evolution, the question was, are they always like two identical chains, two identical polypeptides that interact together? Sometimes, sometimes not. If you think about over time, let's say you have a single gene that produces this protein, and then maybe it's duplicated at some point and it produces another protein that's similar. They may work together, they may not. You know, it just depends. A lot of proteins, if it's a complex protein, then there are going to be several different primary structures involved in making it. Okay? This one, I think it looks fairly identical. I don't know if they meant it to be that way or not. Okay, and let's talk about what a special class of proteins called enzymes. And these are proteins that make reactions happen in cells. Let me go back. I use invertase a lot. You know, when you go to the store, you buy sucrose. And if I take that sucrose back, and let's say I put some sucrose in water, mix it up so that the sucrose is solubilized. So that every single sucrose molecule, which is a glucose and fructose stuck together, is surrounded by water. And I heat that up, what's going to happen to some of that sucrose? Just because of the thermal energy, the occasional covalent bonds are going to be broken, and you'll end up with a glucose and fructose, but not many. Okay? I could take that same container of sucrose, and I can add the enzyme I always talk about, invertase, and within a very short period of time, all the sucrose has been converted into glucose and fructose. And what invertase does, and we'll get more into structure a little bit later, but invertase grabs that glucose and fructose molecule that's stuck together and bends it enough to cause the bond to break, and then releases the glucose and fructose. And so, like, you've got invertase in your saliva. So most of the sugars that we eat are actually sucrose. So you can break those down starting in your mouth.
Is that why it gets like really, like if you chew on something for a while, like it gets really sweet?
Yeah, it could be, yeah. Or you're releasing, actually, in that case, you're releasing the sugars.
Oh, yeah.
So you can get to your taste buds. You know, when you go and you buy, why don't when you go to the supermarket, why do you buy bags of sucrose? Why aren't you buying bags of glucose or fructose? Where's sucrose come from? Anybody know where the majority of it? Sugar beets and sugar cane. It's because it's the cheapest form of sugar. If you buy an artificial sweetener, 99.9% of what you're buying is fructose. Fructose is a lot sweeter than sucrose. So for artificial sweeteners, there's only a few milligrams of the actual sweetening agent in there. They got to have something to carry it, because if you opened up a container that only had 5 milligrams, you'd never get it into your drink. So fructose is a lot sweeter, and they use fructose in there. Okay? Now, let's go over to the rest of these, and then we'll stop. They are catalysts, and one of the definitions of a catalyst is that it's active in small quantities, and another definition of a catalyst is that it is not consumed by the reaction. Remember that? When you took chemistry, you're talking about adding metals, catalysts, or whatever. So you take the invertase, it breaks down the sucrose molecule, you still have the invertase, it can do it again. So the invertase isn't destroyed by the reaction, which means a little tiny bit of an enzyme will just keep working and working and working and working until it breaks everything down. So I can take my container of sucrose, I can put a few invertase molecules in there, at the right pH of course, and come back and all the sucrose is gone. It's now glucose and fructose. Okay? They are reversible. So a invertase protein enzyme may grab a sucrose molecule and it doesn't grab it quite the right way and just spits it out without breaking it apart. They are usually regulated. We have very few enzymes that are just produced day and night. It's a waste of energy to produce an enzyme that you don't need. They are very specific. So invertase only breaks down sucrose. Okay? Invertase does not break down sucralose, which is Splenda, which is sucrose polyester. So just think about that. For every reaction, there is a specific enzyme. Now, there can be more than one kind of invertase. There can be different kinds of invertase, but invertase will only break down sucrose. And that's very important. Though we can fake it out, and that's a really big area of research. Let's make a substrate that looks like the substrate, and we can make the enzyme do something that it really shouldn't do. Okay? So like the enzyme that I keep talking about that you find in the mitochondria, if we give the mitochondria cyanide, guess what? The enzyme thinks that's oxygen and binds the cyanide. So we can use something called competitive and non-competitive inhibitors. Don't worry about that. We'll get that to the next time. But we can fake it out. And then you can think about doing that in terms of disease. And maybe you can cause something to break something down that it normally wouldn't break down. A lot of research going into that, because now we have these, supercomputers, and we know what the 3D structure of the enzyme is, and then we can go in and we can change an amino acid and see what that does to the binding of the substrate. So it's a lot of work going into that. They have very specific characteristics. So there's acid invertase that works around the acidic pH levels. There are invertases that work better at higher pHs. Okay? And the last one is they need cofactors. The reason why we need vitamins is so our enzymes will work right. Without vitamins, The enzymes cannot have the right 3D structure. And cofactors. They need that for the right 3D structure. If we're limited in some of the minerals we take in, or this is especially true for plants, without iron, molecules don't form the right 3D shape, so they don't work well. They don't carry out their reactions. Plants also need cofactors and vitamins. Okay? Questions? We'll stop there and pick that up next time.
Activity is highly dependent on pH and for other stuff, and then you said most active during ambient temperatures in plants. What's ambient temperatures defined as the optimal temperature for that plant?
Yeah, that would depend
That's
on
what I thought
which,
it was, but I didn't know.
that would depend on
Just because you said like some of them need real high, some of them need real low.
yeah. I mean, there are optimum pHs. Oh, come on, I need to get you up there where I can copy you. Good Lord.
Now, for most plants, is